Hello and welcome to the first part of Unit 6 on serology. In this part of the unit, we'll be looking at body fluids. So some examples, blood, saliva, semen, tissue, feces, urine. In particular, we're going to focus on blood and semen. Those are the most common types of body fluids associated with crime. So that's where our focus will be for this first Unit 6 on serology. So as I already mentioned, serology refers to analyzing body fluids in relation to a crime scene. In the past, the body fluids were analyzed for different aspects, but currently the most common species that would be examined for is DNA. So if the body fluid has DNA evidence, then it can be individualized to a single person or down to identical twins if we happen to have that case, but it's a much better and more individualized type of sample than in the past. Blood's the most common body fluid found at a crime scene, and in the past we would just look at blood type that we'll talk about in a moment, but nowadays, of course, we can use that blood hopefully to identify an individual by DNA analysis. So Karl Landsteiner, we talked about him a while back toward the beginning of the course, and he's the father of blood typing. So this work is not all that old. This was around 1900 that he did this work and won the Nobel Prize for it a few years later. He devised the system that we now know as the ABO blood system. So this allowed for the class characterization of evidence in a few key cases, like the Jeffrey MacDonald case. The blood evidence was particularly helpful because it was a special set of circumstances, and so we didn't need DNA analysis necessarily back at a time before DNA analysis was possible. The blood typing itself was very helpful in that particular historical case. So what Landsteiner was able to determine was that certain individuals had blood incompatibilities. So if we were to attempt a transfusion between two individuals, sometimes it would work, sometimes it wouldn't work. And if it didn't work, it could ultimately be fatal because of these incompatibilities. So through his testing, Landsteiner was able to find that individuals fall into one of three groups, A, B, or O, and then later on some colleagues found the AB blood group. So if you have type A blood, that means that you have the type A antigen on your blood cells, and therefore in your plasma you have the anti-B antibody. So if you're an A individual, you can't receive a B individual's blood, and likewise a B individual cannot receive an A individual's blood because where the A individual has the A antigen on their blood cells, the B individual has the B antigen on their blood cells, and each has the anti for the other antigen in their plasma. So the antibodies will agglutinate, they'll clump up with the foreign antigen on those foreign blood cells. If you happen to have type AB blood, then you wouldn't have that problem because you don't have antibodies in the plasma since you have both the A and the B antigen on your blood cells. If you had the antibodies in your plasma, you'd attack yourself, and that wouldn't be a very good thing. So those individuals can receive from either AB or the last group on our slide, group O, which has neither the A nor the B antigen, and therefore has both anti-A and anti-B antibodies in the plasma. An O individual could give to type AB individual because when we give blood in transfusions, we get rid of the antibodies from the plasma. So it would just be the blood cells, and since the type O blood cells have neither A nor B antigens, any of the other groups, group AB or group AB, could receive from group O. So O is the universal donor because they can give to any other type, but of course they can only receive type O blood from others, because if they were to receive A, B, or type AB, then their antibodies would attack those antigens. Group AB would be the universal recipient because they can receive blood from group A, group B, or group O, as well as their own group, group AB. So You may have heard those terms before. Hopefully that's a little more detail on how that all works out. All right, so now looking at the function of blood. Blood is a very important body fluid, and it does a number of important functions. First of all, what we probably know blood best for is a transportation system. So in particular, blood brings oxygen from the lungs to the cells of the body and delivers waste, in particular carbon dioxide, back to the lungs. So that's our transportation system, and that's what we tend to associate with blood is the carrying of oxygen. In addition, in the blood we have chemical messengers, so hormones, for instance, that are secreted into the blood, and they can have effects far down from the body, right? Hormones from the pituitary gland can have effects down in the other areas of the body that they use the blood to get those messengers down to those other regions. Also, nutrients are carried in the blood, and in addition to the carbon dioxide, other wastes that ultimately end up in the urine, for instance, are carried through the blood. Finally, toxins go to the kidneys and then are also excreted through urine or sweat. Another function of blood is to maintain body temperature. We're warm-blooded animals, so we do need to have that warm blood keep our body temperature nice and warm. If you get too warm, then your blood will thin. If you've ever spent a significant amount of time in a warmer climate after having lived in a cooler climate, then once you get used to that, it's because your body has actually adapted through thinning of the blood. The blood also maintains pH, so the acidity or basicity, if you prefer to think of it that way, of the body. The blood has a pH buffer system that keeps it just above 7 and allows for the resistance of pH changes with eating acidic foods, for instance. That doesn't send your blood pH down because your blood has buffers. So if you consume too much acid, like strong acid, muriatic acid, or hydrochloric acid as we chemists call it, we certainly know we can't drink that. We can't drink drain cleaner, right? That's a strong base. That would be problematic because then our blood would no longer be able to maintain its desired pH, and if it goes too low or too high in pH, then it can't do its function. And with those important functions, if the blood proteins precipitate out due to wide pH changes, then you're not going to be able to have the transportation of oxygen and other wastes and nutrients, and you won't last very long. Finally, the last function we list here, it controls the electrolytes, the salts that we know we need for proper body function. So the blood is incredibly important. It has a number of key functions, and that's why we all hope to keep as much of it as possible inside our body all the time. Okay, so how much of that blood should we be keeping in? Well, it's around 5 liters for the average person. It makes up about 8% of our body weight. Again, if you're above average in body weight, then you're going to have probably a little more than 5 liters. If you're below average, then you'll have a little less. But 5 liters is a good average for the amount of blood in the human body. Most of that blood is water. We call that the plasma if we were to separate it out. And then the other components, the key components anyway, are 3 types of cells. The red blood cells, known as erythrocytes. If you know your Greek, erythros is Greek for red. And that's a good thing to keep in mind for the exam. The red blood cells take care of that transportation of oxygen that we talked about for one of the main functions of blood. So hemoglobin is the protein there that helps to transport that oxygen and get rid of the waste like carbon dioxide. White blood cells, leukocytes, again, if you know your Greek or hopefully are learning some Greek as we go, leukos, Greek for white, so leukocytes are the white blood cells, and those are very important because those are the only types of cells that we discuss here in the blood, at least for the mature blood cells, that still have their nucleus intact. So the white blood cells are actually our source of DNA, which allows blood to be an individualized type of evidence because of the DNA of those white blood cells. the nuclear DNA that exists there. They also have an important function, and that's for immune system function. So the white blood cells are key in a healthy immune system. And the other place you've probably seen leuco is if you have heard of the blood disorders like blood cancers such as leukemia. It's the white blood cells that are having the problem there because they have the DNA for the misgrowth of cells. So that's another way to keep the white blood cells straight, is if you happen to know that link to the disorder leukemia. Finally, the platelets are important for blood clotting. They're also known as thrombocytes, and those would ordinarily not be doing too much, but if something happens where there's a cut and the blood starts to leave the body, then the platelets kick into action and start clotting to protect the body from losing too much of that vital blood fluid. So there we see in our image to the right the overwhelming majority of blood being due to the water, and then very small amounts to platelets and white blood cells, although the white blood cells have the particular forensic importance of being the source of nuclear DNA. And finally, again, a little less than half of the blood due to the red blood cells themselves. Okay, back to our friend Karl Landsteiner and the idea of antigens and antibodies. Hopefully your brain's processed it a little bit since I mentioned it before, but let's dive into it again because it does tend to be one of the more confusing aspects of this part of the unit. So antigens are the protein tags found on the red blood cells themselves, and the one we didn't talk a whole lot about before is the D antigen, or the Rh factor for the rhesus monkey where it was first discovered. So we tend to say that you're Rh positive if the D antigen is present on the cells, and you're Rh negative if the D antigen is not present. And about 85% of the human population has the D antigen, and that leaves about 15% to not have it. So plus predominates, but minus is also a possibility. So the antibodies are the proteins found in the plasma, the serum, that 55% of the blood volume there, and those defend the body against the antigens. So if we look at our image, again, these don't discuss the D antigen, but we have the A, B, and AB antigens. If you're type A blood, it's the A antigen that's present on the red blood cells and the anti-B antibodies present in the plasma. If you're type B, then you have the type B antigen on the red blood cell and the anti-A antibodies in the plasma. Type AB individuals have neither antibody because they have both A and B antigens, and we don't want to have a antibody-antigen reaction within a person. Finally, group O have both the anti-A and anti-B antibodies because they have neither A nor B antigen present. So if we have blood incompatibilities here, then we're going to have a situation where if you give a type A individual type B blood, their antibodies are going to kick in and attack it as foreign and cause this agglutination reaction we're going to talk about very soon. Okay, with the D antigen, actually you get one pass. The D antibodies are not usually present at birth for the negative individuals, the Rh minus folks. They tend to get sensitized that first time, and then if they were to get a subsequent exposure, that's where problems could happen. And in particular, mothers whose offspring have a different rhesus factor, those can cause problems. So the mother's often given an injection of a drug called Rogam to prevent those issues from happening later on, because the first birth is usually where the sensitization occurs, and then later births could be problematic if she were not given that Rogam injection. And there's that term agglutination that I've tossed around a few times now, which is just a fancy way of saying the clumping together of red blood cells due to the antibody-antigen interaction. So the antibodies will bind to the antigens. People with type A blood have those type B antibodies. Otherwise, if they had type A antibodies, their blood would agglutinate with itself. So they can't have both the A antigen on the blood cells and the A antibody in the serum, or we would have agglutination and death, so that doesn't occur. Likewise, people with type B blood have the type B antigen on the blood cells and type A antibodies in the serum, because we can't have the type B antibodies, otherwise we'd get agglutination for the same reason we couldn't have A antigens for the type A individuals. If you're type AB, that blood will not have type A or type B antibodies because there's both A and B antigen present, and we don't want agglutination to occur. Type O blood, which is the most common blood, has neither A nor B antigens, and therefore we find that it has both type A and type B antibodies. So the type O individuals don't have the antigens on the red blood cells, but they do have the antibodies for both type A and type B, which is why they can only receive blood from other type O individuals. If you know anything about blood donation, you probably have seen that in the emergency room settings, on the emergency medicine dramas, they're always demanding type O neg, because that would work for theoretically anybody, at least with the major blood groupings, because it's negative, so it doesn't have the type D antigen, and type O, it would not have type A or type B antigen present. So that's what's given in emergency circumstances when there's not the time to type and screen an individual. If it's a situation where the person's already in the hospital, they're having a planned procedure, and then something happens, well, those individuals have already been typed and screened, and so they can likely receive other types of blood as long as they're not type O neg individuals themselves. So as I mentioned in the previous slide, we have the ability to type and screen individuals, which means we can determine their blood type and screen for the most appropriate type of blood that they could receive in the eventuality that they might need to receive some blood. So how do we do that? Well, the way that blood is typed is by making use of those agglutination reactions we just discussed. So if you take that blood sample, then you can add different antibodies, known antibodies, to the sample and see which, if any, cause clumping. So if you take that blood sample, and if we look up at the top, we have a blood sample that's A positive blood, so it interacted with the anti-A antibody, and we see the agglutination. If we take that same A positive blood in the lower image and we interact it with the antibody for type B blood, anti-B antibody, we see we don't get the same reaction because there's no B antigen on the red blood cells there for that type A individual. We don't see agglutination occur. So if we see clumping, agglutination occur with A antibodies, then we know the person's either type A or type AB. The way that we would determine that is we would see what happens with the type B antibodies. If the person has type B blood, it will clump with the B antibodies. So if we saw agglutination with A antibodies and with B antibodies, then the person is type AB. If we saw agglutination with only one and not the other, then whichever antibody gave agglutination would identify the blood type. If we don't see agglutination with either the A or the B antibodies, then we must have a type O individual. And we do a similar thing with the D antigen, right? You add the D antibodies, so if you get agglutination, then that person is positive for their Rh factor because they have the D antigen. If you add the D antibody and there's no reaction, then the person is negative because they do not have the D antigen. And so this is all known as forward blood typing, where we take a blood sample and test it against known antibodies. And so here with that direct blood typing or forward blood typing example, we see a series of results. So if the person is O positive, then they're going to have no response to the anti-A because they don't have A antigen, no response to the anti-B because they don't have B antigen. They will have a reaction with the anti-D because, as we say in the slide, since it's the very top case, they're O positive, therefore they have the D antigen, so they will agglutinate with that. And then the control would just be there as a check to make sure that the test is working well, and the control should not have a reaction. If the person's O negative, then that's the case where we should see no reaction whatsoever because the person does not have the A antigen, the B antigen, or the D antigen, and therefore the control, as always, should be no result. So we would see no agglutination reaction for an O neg individual, and that's the blood that is very valuable because it can be given to anyone because it doesn't have any of those antigens. Right? The antibodies exist in the person's serum, but those are cleansed before the blood is donated, so we don't have to worry about those. They get washed away. If you happen to be an O neg individual and the Red Cross knows about you, then they will call and call and call. They probably come to the house of the O neg individuals because as an O positive donor myself, I know I get those calls as soon as I'm eligible, and they don't stop calling until I make that appointment. So I would imagine they're even more aggressive for that O negative blood because it is the only blood that they can give in a true emergency where they don't know the blood type of the individual needing the blood. Moving on down our list, the A positive case, they're going to get agglutination with anti-A because they have the A antigen, and anti-D because if they're positive for the Rh factor, they'll have the D antigen as well, and then no agglutination for the anti-B or the control. A negative individuals would just have the A antigen present, so they'd only have agglutination with anti-A. B positive would have agglutination with anti-B and anti-D because they're positive individuals and they're type B individuals, so they would have the antigen for B and the antigen for D. B negative would only have agglutination with the anti-B antibodies because they'd only have the B antigen present. AB positive folks would have three results, right? Because they have the A antigen, they'd agglutinate with anti-A. They have the B antigen as well, so they'd agglutinate with anti-B, and they would agglutinate with anti-D because they're positive for the Rh factor. So the only test that wouldn't be agglutination in that case would be the control. Finally, AB negative individuals would have agglutination with anti-A and anti-B only because they would not have the D antigen. They would have just the B and A antigens. Finally, the test is invalid if the control is an agglutination reaction, whether or not the others are agglutination or not. So there we see a problem. Okay, and hopefully that gives a little more visual guide as to how this forward blood typing is done. Reverse blood typing also exists, and that's where you could take the serum of the individual instead of testing their blood cells, test their serum for antibodies by testing them against antigens. So the forward, we test the blood cell antigens against known antibodies. The reverse, we can test the blood serum to see what antibodies are present and then infer the antigens from that. And so now we see a slide on that indirect or reverse typing. This is the method developed by Leon Latz back in 1915. We mentioned him earlier in the semester as well, so he's back again for us to appreciate. And so it's the absorption elution method where the fabric with the blood stain was put into a saline solution to rehydrate it. The antigens were added, fabric was washed, added to another saline solution and heated so that the antigens on the fabric will dissolve in water, and then the known blood cells are added looking for agglutination. So here we're looking really for the presence of the antibodies from the blood serum of the original individual, and then when we add those known antigens, we can determine what antibodies were present. So if the person had anti-B, then when we add the B antigen, we'd get agglutination, and that's how we would know that the person must have been either type A or type O if they had the anti-B antibody present in their serum. So the way we distinguish between type A and type O is we would add the type A antigen, and if we see agglutination, then the person must have had anti-A and they would have been type O. If we don't see agglutination, then they must have been type A. So that's the sort of method that could be used for indirect blood typing, or reverse blood typing as it's now called. Now, paternity testing used to involve a lot of those blood testing principles, mainly to exclude individuals for blood type. But nowadays, it's much more common to just perform a DNA test to identify paternity after perhaps an initial blood screening to see if it's worth the added time and expense of a DNA test. But all of it relies on the fact that mother and father pass on genetic traits to offspring. So heredity is the passing on of those traits. Genes are composed of DNA. They're segments of DNA that code for protein synthesis. So each gene has two alleles, and one came from mother and one came from father. So that will give rise to what we call the genotype. It'll tell you which alleles are really present, and that you would need to actually analyze DNA to know genotype for most things. The different alleles, again, came from each of the biological parents, one from the mother, one from the father. They can be dominant, where they're always expressed. They can be codominant, where they're both expressed. They can be recessive, which means they're only expressed if both alleles are the same. And we can also give rise to terms that you may not have heard before, but hopefully you're familiar with what it means to be homozygous, which is the same sort of thing as recessive, right? Where both alleles are the same, but it doesn't have to be a recessive allele. It just means they're both the same. So a recessive individual will always be homozygous. Heterozygous means the alleles are different, and the phenotype is the expressed trait. So blood is an ideal example of these different features, because blood has that idea of dominant. A and B blood type are dominant. You have codominance presence possible, so you can have type AB blood where both the dominant alleles are expressed. You could also have a recessive case, which is type O, where you have neither A nor B, and you only have type O when you have neither A nor B present. So for homozygous case, type O blood is always going to be homozygous. Type AB blood is always going to be heterozygous, because the only way you get type O is if both mom and dad gave the O allele, which is the recessive trait. Just because it's recessive doesn't mean it's uncommon, right? Type O blood is the most common blood, but it's also the recessive trait. So it just means you have to have both recessive genes, one from mom and one from dad. Doesn't mean it's not common. It's the most common blood type. Heterozygous, in this case, the type AB blood is always going to be heterozygous, because you got an A allele from mom or dad and a B allele from the other parent. And that's a case that's actually quite rare. AB blood is the least common blood. So those are the two that we know. Phenotype, which is the expressed trait, if you have type O blood, you have that phenotype. If you have type AB blood, you have that phenotype. Where things get interesting is if you have type A or type B blood, it's sometimes the case that you have type A blood because you have an A allele from mom and an A allele from dad, but oftentimes it's that you have an A allele from one parent and an O allele from the other parent, and because the O is a recessive trait, it does not express. So phenotype can be determined by the simple blood test, but to know the genotype, to know if the person is type B blood, for instance, because they have two B alleles or they have a B and an O allele, can only be determined by genetic testing. So type A or type B blood can be homozygous, where you have both A alleles or both B alleles, or it could be heterozygous, where you have an A allele in the type A case and an O, a recessive trait allele, or for the type B case, you have a type B allele and a type O allele. So the phenotype in either of those cases, where you have type AO or type AA, or in the type B example, type BO or type BB, the phenotype's the same, your type A or type B blood, but the genotype could be different depending on if you're homozygous, both type A or type B alleles, or heterozygous, a type A allele and a type O allele, or a type B allele and a type O allele. So one of the ways we do this is to do what's called a Punnet Square. So hopefully you've done these before, probably in middle school or high school biology class, you've done Punnet Squares. So the example here is awful because actually eye color is a lot more complex than this, but if it were true, we'll pretend that it's this simple. If we had a capital G as the allele for green eyes and a lowercase g as the allele for brown eyes, then we'll consider that capital G green eye allele to be the dominant and the lowercase brown eye allele to be the recessive case. And we see if we had a heterozygous mother and heterozygous father, so they each have a capital G green eye allele and a lowercase g brown eye allele, then we see if the two capital G alleles come together, then you'll have a homozygous green-eyed offspring. If the capital G from one parent pairs with a lowercase g from the other parent, then you'll have a green-eyed individual for phenotype, but they would carry the brown eye allele. If we move down to the second row, now we have the lowercase g allele from that parent pairing up with a capital G allele from the other parent for the first case. So the first box in row 2, the same as the second box in row 1. And then finally, we could have both the lowercase g from the mother and the lowercase g from the father pair to give the recessive brown-eyed eye color in this example. So we see the phenotype, the offspring having green eyes, would happen 3 out of 4 times, right? The one case where it's a phenotype that is a homozygous dominant green-eyed offspring, and the other 2 cases where it's a heterozygous offspring that still has the capital G dominant green eye allele, so they would express that, we assume, again, from this grossly oversimplified example. And then finally, 25% of the time we would have brown eyes expected because that's the case where the 2 lowercase alleles occur, and we only get that 1 out of 4 times. And we would know both the phenotype and the genotype for the recessive. For the other cases, we wouldn't know if we got the capital G or capital G lowercase g unless we did the genetic testing. So that's an example of Punnet Square, and hopefully it's bringing back memories of something that you've done before and are hopefully fairly comfortable with. If not, please do reach out for some guidance if it's something that causes you to struggle a bit. All right, so let's see how well you've remembered this skill or developed it if you didn't learn it back in your prior education. So for the following example, let's determine the phenotype. A father's blood genotype is homozygous BB. It's very rare we would know that he's homozygous, but for this problem we'll go ahead and say it. And a mother's blood genotype is heterozygous AO. What percentages of phenotypes would their offspring have? So go ahead and pause the video here while you work on it, and then when you're ready, we'll come back and we'll figure out what, hopefully we'll see that we agree, or figure out what may have gone wrong if we don't agree once you've started the video back up. Okay, so in the bottom of the screen, hopefully you arrived at a similar, hopefully the same actually, Punnet square. So the A from mom and the B from dad could pair up as AB for either of those B's that dad gives along with mom's A. For the O example, mom could give her O to pair with either B from dad, and we get a heterozygous type B offspring. Actually, this is the most common way that you would find out that you're heterozygous yourself. So the mother here might not have known she was heterozygous, but if she gave rise to one of those type B children without a type A antigen present, then she would know that that's from her having a type O recessive allele along with her A allele. So 50% of the time we'd expect this parent group to have type AB offspring, and 50% of the time we would expect type B phenotype. It would happen to be type BO genotype. So we would know that as well just because of the fact that we had a type O mom who did not give rise to a type O child. Now, in terms of the mother on the side and the father on the top, that's arbitrary. I consulted a biologist and she told me it doesn't really matter who goes where. Of course, we get the same outcome either way. So I guess there's no real convention about it. Just whoever you want to put on the side, you put the other parent up on top. So hopefully that went well for you. If not, like I said before, please do reach out for some help if this is causing you any trouble. Okay. Otherwise, we'll go ahead and move on. So we've discussed how valuable body fluid evidence is. Blood we've just been spending quite some time on, and of course the white blood cells in that blood can be sources of nuclear DNA, so those can individualize that evidence and make it very, very valuable indeed. So since blood is the most common body fluid found at a crime scene and blood has the potential to be individualized evidence, let's talk about how we go about examining what we believe to be a blood stain. So there's some preliminary or presumptive, if you want the official term, tests at the crime scene. First and foremost, the test need to determine, is it blood? So not every red stain is blood, and we need to be able to identify fairly quickly if we're likely to be dealing with blood or if we're likely to be dealing with something else. So these tests rely on the presence of hemoglobin in the sample, the hemoglobin that we mentioned earlier as being the oxygen carrier of the blood. So there's the Kastle-Meyer phenolphthalein test, there's the hemostick benzidine tests, and so the Kastle-Meyer phenolphthalein turns a deep red, deep pink, magenta, I should say, in the presence of blood, whereas the hemosticks, which is the benzidine-based test, turn green or blue-green in the presence of blood. So if we get any other result, then we're likely not dealing with blood. If we get a positive result, then we're likely dealing with blood, although there are sometimes false positives, but that's why we would follow up with a confirmatory test if our presumptory test is positive and we have any reason to question it, or if we get an unusual result later on in our testing, we may find that we had a false positive. We do have some other tests, and hopefully I forgot to mention it on the previous slide, but you should go ahead and click those other tests that we talked about, the Kastle-Meyer and the hemostick test, to see a little video of the test in action. Some other tests that we don't have video links for are the luminol and blue star. Again, luminol was largely replaced by the blue star method, which relies on the same sorts of ideas. So in either case, the chemical species is sprayed and glows. You have to be in the very dark for luminol, not quite so dark for blue star, which is why, as I said, it's largely replaced luminol. So these are very, very sensitive tests. Blue star is the most sensitive, can detect very little amounts, even when areas have been cleaned. So most suspects, if they know what they're doing and are trying to cover their tracks, have attempted to clean up after themselves. Blue star, if you spray a room, you're likely to find the presence of very small amounts of blood. Even if the person has done a decent job of cleaning up, they likely haven't gotten quite all of it, and blue star is sensitive enough to find it. Fluoresceine's another test. Again, it's sprayed and then a UV light source is shown upon it, and if you see that UV glow, then there's likely the presence of blood there. Finally, chemical enhancement can be used to enhance a blood stain to make it easier to see, get a blue or a purple blue color that again brings out that blood stain that may have been difficult to see without that chemical enhancement. And so what our text calls confirmation tests, you're more likely to hear the term confirmatory tests used. These are necessary because many of those presumptive tests that we mentioned just a moment ago can give false positives, and so you need something to follow up and say, yes, this is definitely blood, and that's what a confirmation or confirmatory test is all about. Generally, the reason we do the quick presumptory tests is because they require very little sample. They can help you find that sample in a crime scene that you don't even necessarily notice that there's blood. The sensitive tests like the blue star test can help you find that. You do need larger samples generally for the confirmatory test. So the Teichmann test reveals brown crystals in the presence of blood, so that would be evidence that you do indeed have blood. And again, the presumptory test may give false positives, but the Teichmann test should not. The Takayama test is the more common. It's the most commonly used confirmatory test. It's an update to the Teichmann test, and it reveals pink crystals in the presence of blood. And again, these tests have high confidence. So if they give these results, then you are definitely dealing with blood. You don't have to worry about false positives here, and that can be reassuring to a jury if you're presenting blood evidence. So once we've confirmed that we are dealing with blood, the next question is that human blood? So we don't necessarily want to jump to conclusions. If we find blood in a kitchen, that's not terribly unusual, right? Those of us who consume animal flesh and prepare that raw meat in our kitchens are likely to have trace amounts of blood from chicken or beef or pork. So it's important that we do our due diligence and determine if indeed we've found that we have blood, is it human blood? So the precipitin test is the usual method there. So you add the sample and human blood antibodies that will only react with human blood to a test tube. If you get a precipitation, so a band or ring of precipitate will indicate that it is human blood. If you don't, then it was blood, we know that, but it was not human blood, and then we could run a test for various other common animal bloods, like those animals that I mentioned that are food for many of us. So the specificity of antibodies is really, really high. That's what we talked about earlier about blood. So the antibody test is quite sensitive, and so if you don't see that precipitation band or ring, then you don't have human blood, and you would need to test for the other animal blood if you wanted to find out what type of blood you have. Another method is gel diffusion. So you add that sample and the human blood antibodies to a gel plate. If they migrate toward each other and form a precipitate, then you're dealing with human blood. If that doesn't occur, then just like a negative precipitin test, you are dealing with blood that you know already, but it's not human blood. So let's presume that we are dealing with human blood, and now we're looking to determine whose human blood we have. So the way we would do that is by DNA electrophoresis. So this is a method that will allow us to separate DNA based on size by the use of an electrical current, and we would have used some sort of chemical agent to impart a charge among these different fragments of DNA, and so the smaller fragments would move further distance toward the charge. Larger fragments would move more slowly toward the charge, and we can separate based on size in that way. If we've stained them with some sort of fluorescent tracer like you see in the right hand image, then after we run our gel, we can image our gel and determine which bands we have, and ultimately compare those to known samples that hopefully we'll be able to require the suspects to submit, and you can figure out which suspect you have the DNA for based on whose banding pattern matches the pattern from the scene. So now moving away from blood into the other body fluid that we'll discuss here in this unit, semen. So again, there are other types of body fluids that can be important at a crime scene. Blood's your number one and semen's your number two. So those are why we focused on those two. But again, I don't mean to discount the other body fluids, but for the time's sake and the purpose of an introductory course in forensic science, we'll just focus on those two examples at this time. So semen itself is about 95% seminal fluid, which is produced by the prostate, and then 5% or so sperm or spermatozoa produced by the testes, at least for those males who have sperm present. We'll talk about that in a moment. So sperm is very fragile. It's the carrier of that genetic information. So sperm meets egg, and that's how you started. That's how all of us started life. And it's, again, very fragile, so they become brittle and dry. And some men produce either very little oligospermia or no sperm, aspermia, where they have absolutely no sperm in the semen. That could either be, again, a natural phenomenon, or aspermia could result from vasectomy if the male has voluntarily or involuntarily undergone a procedure where the vas deferens has been cut and tied from each testis. Then if that's been done to both testes, the man will have no sperm present in his semen. So that's why we can't just look for sperm, because it's possible that the assailant had no sperm present. So we really prefer tests that identify the seminal fluid by something other than sperm. Okay. Oftentimes finding those seminal stains is not too difficult. Most of the stains are visible. You're looking for an off-white to yellow, crusty appearance on a dark surface. If it's a light surface, you might need a blue light or an ultraviolet light to see those stains fluoresce. But again, it's not usually terribly difficult to find those stains. If you want to sleep a little better at night in hotel rooms, you probably want to leave the ultraviolet lights at home because these particular stains can persist even through the wash. So yeah, those sheets are clean. I'm sure they've been laundered, but if it helps you sleep, then club soda will also fluoresce. So maybe someone spilled a little club soda on those sheets. So if we have what we believe to be semen stains, of course, like any body fluid, we would want to package that evidence in paper bags to allow it to air dry. We want to have, again, a non-airtight package so that we don't build up those environmental molds that could degrade our sample. The earlier, quicker the sample's taken, the better the results. Again, this would be for someone who unfortunately underwent a sexual assault. The victim, we would want to have that victim come forward as quickly as possible because, again, the sperm cells will begin to break down. Our textbook, assumes that it was a male raping a female, but it could be, again, a male against male, and then we'd be looking other places, but the same sort of things happen with the sperm breaking down. Sometimes we can't recover sperm at all because the the assailant may have aspermia, right? They're aspermatic, they have no sperm, or oligospermia, they have so few sperm that it's tough to find any, or they've had the vasectomy that we mentioned, or the sperm themselves are broken when they're being collected, right? Collection usually occurs with rubbing against the semen stain, and it's possible to break those fragile sperm, and that may be another reason those sperm cells are recovered. So we want to try to collect the sample early so that we can have our greatest likelihood of finding sperm in the semen if they're present. So like we were looking for blood and trying to confirm the blood being actually blood first and then human blood second, we need to perform some tests on the semen to determine if indeed we're dealing with semen. So the first test is the brentamine spot test, and this is a test for the acid phosphatase from the prostate gland. The seminal fluid has it, but it's not specifically from sperm cells, which again, maybe there's no sperm present anyway, based on those conditions that we talked about. So this test would give a positive purple result in 30 seconds or less, but it has some drawbacks, and those are the false positives. So vaginal secretions can give a false positive, some contraceptive creams, even watermelon could give a false positive, but again, all of these would take much longer than the positive result for semen. So it's still a good test, but it does have its drawbacks. MUP tests also for acid phosphatase like the brentamine, but it's more sensitive. Christmas tree stain, that's a test that confirms the presence of sperm cells. The heads are red and the tails are green, which is why it's a Christmas tree for the name. But again, it's possible that the semen doesn't have any sperm in it if the male's aspermatic or oligospermatic or has had a vasectomy. So this may not be appropriate just because you get a negative result here. Likely you would want to follow up with something like Abicard P30, so the prostate-specific antigen test. This should be a positive result for, again, semen, whether or not sperm are present in the males out there. Once you get to a certain age, you'll be following your own PSA levels, and as we mentioned before, the seminal fluid is about 95% from the prostate gland. So when males do have abnormal prostate-specific antigen levels, indicative of perhaps of prostate cancer, they typically don't do too much. It's usually a fairly non-aggressive cancer, although it can be aggressive. So males tend to just wait and see, whereas females, if they get a breast cancer diagnosis, for instance, they tend to be a lot more aggressive about treatment. But males realize that there'll be some definite dysfunction if you're removing a gland that's responsible for 95% of that seminal fluid. There's other positives, false positives with the prostate-specific antigen test. Actually, believe it or not, breast milk will have small amounts of PSA in it. So again, it's usually easy to tell that it's a false positive because it takes a long time. It's not very intense, but that is a drawback. Finally, the rapid stain is actually a very, very good test because it tests for semenoglin in seminal fluid. So that's specific to seminal fluid. You're not going to find that in breast milk. It also doesn't rely on sperm, which may or may not be present in the sample. So it's a really nice test, and actually a really good way to confirm that you're dealing with semen and not some other body fluid. I mentioned a little bit when we were talking about analyzing the semen from what we believe to be semen stains at the scene, that time's very sensitive because the sperm breakdown and all those things. And I brought up the sexual assault issue. So just to follow up on that, just like timing is critical for the collection of semen stains at the scene, timing is critical for collecting the evidence from the victim as well. So a forensic medical exam would be performed on a sexual assault victim at a hospital setting. So they don't have this done at the police station. They go to the hospital for a medical exam, a forensic medical exam. The victim will stand on a sheet. The sheet's there to catch anything that might fall. They remove their clothing. The clothes are placed separately into paper bags. Samples will be taken from the head and pubic hair, swabs from vagina, cervix, rectum, mouth, whatever is relevant. Head hair, blood, fingernail scrapings, urine, and saliva residue from the skin will be collected. And again, it's a very, very terrible thing that's happened to the victim. They generally will feel dirty and guilty, and they'll often want to clean themselves, take a shower. It's the worst thing that they can do, but it's a very understandable response. So hopefully the victim is aware enough that they come right to the either police station or the hospital to be analyzed for physical evidence from the sexual assault, so that there's as much evidence as possible. But yeah, in the past it's been very unfortunate. People have been shamed and things like that. I think that's far less common today. I think victims are really treated well and believed, and so hopefully they feel comfortable coming forward and know that they need to come as soon as possible so that the evidence can be preserved and the assailant can be brought to justice. And finally, another body fluid that can be a great source of DNA is saliva. So saliva can be present at the scene. Cigarettes, if the suspect's a smoker, saliva might be present on cigarettes. Gum, if they're a gum chewer. Bite marks on an object or a person. Stamps are less and less likely, right? I think pretty much all the stamps now are self-adhesive, but in the old days you had to lick stamps. I think some of the extra postage stamps may still require that, but that used to be a relevant thing. That's not so much anymore. Envelopes, at least some of the time you still have to lick the envelope, but there's a lot of self-sealing envelopes as well where you just remove a covering and it's already got an adhesive there. Whatever the case may be, you'd want to let the evidence air dry first, then package in a non-airtight container. A paper bag is your best bet. And sometimes you can get the blood type from the body fluid, right? So secretors are people who secrete blood antigens into their other body fluids, saliva, urine, sweat, and so you would be able to tell blood type from that. But more commonly, you're going to use the saliva for nuclear DNA. Hopefully that's present, and then you can figure out the individual rather than just their blood type. The other time you would use what we see there in the image, that's a buccal swab. The person is swabbing the inside of their cheek with a cotton swab. That's typically how a DNA sample is taken. So whether it's a DNA sample that has been court ordered or something you want to do yourself, if you want to go and have your DNA analyzed for one of these genetic testing kits, it's usually a buccal swab, which involves those cheek cells that are caught in the saliva. So that's another example of how you might be analyzing saliva evidence if someone's been forced to submit a buccal swab as relates to a particular criminal investigation. So there we have it, mostly blood, a fair deal about semen, and then finally just a bit about saliva for our foray into serology. Hope you enjoyed it, and we'll come back and talk about blood spatter in our second part of Unit 6 on serology. Hope to be talking to you again soon. Take care, everybody.